All right, guys, I am very excited for this conversation. Dr. Himel Patel, he is actually nearby me. I reached out to him after listening to his interview with Ben Greenfield and Chris Beers on mitochondria and how we can test our mitochondria function. So, Dr. Patel is the chief scientific advisor of Versailles Discovery, which is literally, I think, 10 minutes from me. So I need to get over there. They are the creators of what we'll talk about today, mescreen, MESCREEN. If you are listening and not watching the video, he is a tenured professor and vice chair for research in the Department of Anesthesiology, University of California, San Diego, and has a dual appointment as VA research career scientist and VA research pharmacologist, VA San Diego Healthcare. He obtained his PhD in Wisconsin, the medical college there, in pharmacology and toxicology, and did postdoctoral work at UCSD in the Department of Pharmacology. So, how did he get towards doing a mitochondria scan? We'll learn about his journey and ask him his story, as I always love to learn more about what drives people. What is their why? What is their purpose, their mission, and passion? Dr. Patel has made contributions in shaping new investigation areas and defining the impact of membrane structure on cell, organ, and organismal physiology and metabolism with implications for cardiac. You can read the rest, but for health. So just really look forward to this podcast talking about the Me Screen, how it works, is an at-home collection, analytical process. They give personalized recommendations, expert advice. So I would love to see how I can work with them. Easy at-home testing procedure. You get the kit, register it. Step 2, digital questionnaire. Step 3, collect blood sample and submit to their lab. No more than 10 minutes to perform. Collect everything. You ship your sample. And I've not done this yet. See pictures on the left if you're watching the video. If not, if you're on the audio, I would go to YouTube channel and hit subscribe to Coach Debbie Potts show, where all the podcasts are posted first on video. Now receive your samples little 3 to 4 weeks. And then the Me Screen results include mitochondrial efficiency. And I want to ask the definition on here as we do this with my Pinot metabolism testing. What is the efficiency? Write that down because I had all these terms I wanted to ask. How would they describe it? Efficiency, inefficiency. What do we learn from this test and how can we improve our mitochondria? And of course, I always like to ask, why do we need to improve our mitochondria as we're aging? And how, looking at how to be fit and healthy from the cellular level, what can we do from testing and not guessing our mitochondria function and avoiding dysfunction? So interesting information here. We'll ask Dr. Patel about it, but how you get personalized insights to support your mitochondria health. So testing and not guessing again on what to do. As I keep talking about, do we need to do this or do you need to do that? Instead of being stuck in the cycle trial error, really figuring out what we can do with this data. And if we can ideally collaborate and do Pinot metabolism testing and see how your mitochondria work at rest and during exercise at various intensity levels. So here he is. Let me bring him in. I'm going to push pause and come back to you. Okay, I love talking about mitochondria, and I found an expert, and we are going to dive in deep, maybe go down some of my typical rabbit holes. So, Dr. Patel, thank you for coming on my podcast to help us learn what to do to be fit and healthy from the inside out at a cellular level, those mitochondria, to improve how we age.
Perfect.
So, talk about, I just was reading your intro, your bio, and I'm interested to hear kind of how you got to where you are now, focusing on mitochondria. What brought you to that being your passion?
Yeah, I mean, I've been in this space since grad school. So my PhD thesis was focused on how to protect the heart from a heart attack. And it turns out the heart is one of the major consumers of energy in our body. And the way you protect it is you protect that energy, right? And so, what most people don't realize is that the injury in the heart happens during this reperfusion phase, right? So, you have a heart attack, so blood and oxygen and nutrients and things
Mhm.
stop getting to the critical cells that are maintaining functionality. If you stop that blood flow long enough, you will die. But typically, what happens is you go into someone does CPR on you, or you go into a hospital setting and they bring the blood back. This is when most of the injury happens. It's because your mitochondria don't function well because they've maladapted, right? And so now they see this flood of nutrients and oxygen, and they don't know how to handle all of it, and they make free radicals, and energy is compromised, and you get damage that occurs. And so we've been trying to figure out how to fix mitochondria since those early days. I'm going to date myself, I guess. I graduated with my PhD in 2002. And so over the last 20 plus years, this has been a focus of what my sort of brain's interest have been. I came to UCSD to do my postdoc, and at UCSD, the focus really shifted from heart-based things to more vascular kinds of things that we were looking at. But this idea of energy and stress adaptation continued. And so I became a membrane biologist. And so now I really look at how the membrane is a stress adaptive part of the cell. And it turns out that the membrane has this unique relationship with mitochondria in the cell, and it's that relationship that really allows for stress adaptation. Um, as we started sort of building the lab here at UCSD, one of the big things we did is we created lots of tools to measure mitochondrial functionality in cells and in rodent models that we were looking at, and then ultimately in human clinical studies that we were doing on campus. One of the limitations of those studies has always been scale, right? And so the gold standard in this space is a muscle biopsy. Who wants to come to a facility like ours, have their leg numb to get a giant needle stuck in and then get
Mhm.
some of that muscle out? And it takes us hours to then analyze that tissue in really fine detail to figure out what your mitochondria are doing. It's an amazing tool to have, but it's research centric and very research limited. And so what we wanted to do is develop something that would give any consumer, anyone out in the world, access to their mitochondrial functionality without having to go through this traumatic experience of a muscle biopsy. And so what we did is we started thinking about easy captures, and blood becomes an easy capture. If you have something wrong with you, you go to your doctor, and one of the first things they'll do is a blood test. Um, there are blood and urine tests out there that claim that they can measure mitochondrial function, and they're biomarker based, right? And so one of the things that I think about these biomarker tests, they're the potential of something that may or may not happen, right? It's not an actual measurement of something that's happening, but you're trying to infer based on one or two factors that you're measuring. It's not taking the global system into place. And so we had this, I've always been in these weird lucky places at the right time in my career in life. And so NASA was doing the twin study where the twin went up into space for a year and the ground twin. UCSD was picked as one of the 10 sites to do certain data analysis around these samples. My good friend Brenda Rana was the site PI at UCSD. She knew all the stuff we were doing in the lab. And so she said, "Oh, NASA's very interested in mitochondria, so could you help us develop some sort of way to measure mitochondrial functionality?" The caveat is we can't give you anything other than plasma. And so what we came up with is that plasma is a biochemical capture of who and what you are, right? It has your metabolites, your exosomes, your proteins, everything that your genetics, your microbiome, your disease, your health, everything that's being created in your body gets captured in that plasma. With these biomarker tests, you're looking at one or a subset of these factors. What we wanted to do was develop an assay that looks at the entire you. And so we basically adopt you onto a cell that's never seen you. And we then look to see how that cell's behavior changes. And we can basically reflect what's happening inside your body with those certain things. For NASA, we essentially built a human on a plate assay, and we could figure out different organs at different times of space flight where they would decline. And so this was the underpinning of mescreen. And so then the scalability of mescreen is you with the blood draw, you'd still have to go see someone, right? You have to go to a Quest or LabCorp and get a draw. We've miniaturized this now to a blood card. And so it's literally 4 to 6 drops of blood. You get a kit at your home, you get a lancet, you poke your finger, you drop blood onto this card. The card separates the serum out. It solves the cold storage problem as well. The card is a dry blood card. We've shown stability at room temperature out to 2 months. And so you could literally get a sample from anywhere in the world. It comes to our facility in San Diego. We punch the serum out. We reanimate it in our proprietary buffer, and we're off to the races and can run essentially that same type of adaptive assay that we were doing for the high-end NASA samples that we had. And so it really gives you a very dynamic measure of the actual functionality of what your mitochondria are likely doing. So,
It's amazing. And why this, there's so many reasons, but what are the top reasons this is so important to test?
well, so I get this question a lot. So who should take this test? If you're living and breathing, you should know what your mitochondrial score is, right? Whether you're healthy
Yes.
or not. It's a number that people haven't had access to, and so they haven't been curious about it. But it really does give you a sense of what your global state of metabolism is. And it can, and one of the things we're seeing is people who feel completely healthy or completely sick that have run other tests that don't really show markers. It turns out their metabolism, their energy, really is a good indicator of how healthy or diseased they actually are. Um, we're not a diagnostic, it's not a diagnostic disease, but it gives you a global sense of your wellness, right? And
Mhm.
so it really does give you a target of whether you're achieving what you think you're achieving, or could there be places where you could improve and change a few things to change that number in a unique way.
Hmm. That's why I think it'd be so interesting to see how the results are compared to a metabolism test. I was saying I do own the Pinoe equipment, but a resting metabolic rate when people are being measured their oxygen and carbon dioxide
Yeah,
out to
yeah.
see the correlation, I think would be fascinating. And then doing an exercise test we call active metabolism. How do your mitochondria function at low level to higher levels of intensity and what changes to really
Yeah,
see?
I mean, that would be an interesting comparison. One part of the test does look at a stress response in a dish. And so we can
Mhm.
actually make the cells exercise in a dish, and we can see how your biochemistry either adapts or maladapts against that stressor, right? And
Mhm.
so it'd be an interesting comparison with your protocol and ours to see where people stand essentially, right?
What other lab work do you use to correlate against the blood work that you're doing? You're doing, not just one blood drop, it's six, say 5 to 6 drops, and then
Yeah.
comparing that or adding that in to correlating with other functional lab testing or blood
Um,
chemistry
I
markers.
mean, we don't, right? We've done validation studies where we've looked at metabolites and we can see that certain things go up and down, that they're reflective of what's happening. But again, it's when you start limiting the things you look at, you bias the look, right?
Mhm.
And this is what most diagnostic tests will eventually do is they'll fixate on one or two things that are then predictive. Some are very predictive of this, but others may miss the larger picture of what's there. And so our test sort of is biomarker agnostic in the sense that we don't really care what you're expressing. We're
Mhm.
seeing how the whole you, when we transfer it to the system, creates this effect.
Mhm.
The leap then is, well, how do you know that my blood is creating this biochemistry and this change in my mitochondrial functionality? So we've done validation studies in animal models that we can have easy access to muscle biopsies and blood. We're in the process of doing this in human samples as well. And so there's lots of ways to validate this out. The other aspect that we see with this biochemistry is not something you can change very rapidly, right? We're looking at something that's more of a comparable to an A1C versus a glucose measure. It's not something that dynamically shifts minute to minute, hour to hour. It's really something that when you targetedly live your life the way you do, is a good barometer of where your status is. And then it should really take you a couple months to really change it in a dramatic way to then reflect what happens to your overall mitochondrial score.
It'd be amazing if doctors would use this in the medical setting, as my dad had a heart attack and then passed away a couple of years ago now, but just the impact of how mitochondria plays a role in that after you have a heart attack. And he was in the hospital for weeks, and he would
Yeah,
say 15 years later, he ended up with kidney issues and I think liver issues, type of cancer. But it's just so amazing to look. I was looking backwards. Why do all these things happen? And how
yeah,
is this related to how we're performing at a cellular level, specifically
right.
mitochondria health?
Yeah. Yeah, we're
Hmm.
starting to think about, as the company sort of grows, we're thinking of non-dilutive funding to actually go after diagnostic testing. And so I was just at a conference last week, and one of my good friends, who's an anesthesiologist at Stanford, we started talking, and I was describing him this test, and he's like, this would be really cool in sort of the surgical setting, right? That there are certain patients that come in to have specific surgeries, and they can be really old, but they're fit and they do really well in the surgery. And there's other people that are younger that look like they should do amazing in a surgery, yet they have all these complications that come up. So could it be this energetic state that they're in? And could a
Mhm.
test like a mescreen actually allow us to see if someone is an ideal candidate for surgery, whether they got other things that need to be managed at a basic state before they move to that sort of more stress state kind of thing as well. And it is likely possible, right? And so we are thinking along those paths, and we'll eventually go down that route, which would be great
Hmm.
to see.
So go over the role of the mitochondria. Everyone always says, powerhouses,
Yeah.
they create their energy currency. How else would you explain the importance of mitochondria, as we often relate it just to muscle, but talk about entire body, how it
Yeah.
is an important role?
I mean, they're in every cell of your body except your red blood cells, right? And so they are truly the vigilant organelle, I think, that is measuring and somehow spying on all the energy you're generating in all your organs. The other aspect, how I think about them, is if you think about their origin, right? They were an old bacteria. They got incorporated into a cell probably 1.5, 2 billion years ago. And they gave us a competitive advantage in a unique way. It allowed that cell to create a lot of energy for less resources. And that energy then turns into complexity, growth, other things. As we've sort of evolved, these mitochondria have evolved with us. And I think that they become this conduit, right? One of the big sort of spaces in science right now is the gut microbiome.
Hmm.
There's a gut to everything axis. People think that this may be your real brain. And so the other thing that we've been thinking about is if your gut's going to talk to something in your body, it's going to talk to something it knows. And bacteria look just like your mitochondria, right? And so there is that relationship. So we think that there's this exchange that likely happens across the body in that space through metabolites and other features that communicate that energetic signature. The powerhouse thing, I think, is what you get taught in, I think, kindergarten now, right? It used to be high school or grade school, but then it's younger and younger. It's true, right? They are the energy making component of the cell, but they do so many other things, right? One of the other things that we think about is the origin of how we get our mitochondria. So most of our mitochondria come from our mother. And so you can actually track back, if you were to look at mutational sequences, the first Eve, mitochondrial Eve as well. And so there is this unique feature of genetics and other things that tie into mitochondria. Um, a good way to illustrate the unique features that mitochondria actually create in living systems would be the heart, right? So heart is about 30% by volume mitochondria. So it is literally a bag of mitochondria. You generate anywhere from 4 to 6 kilograms of ATP a day, yet you don't gain that weight, right? In a day, you don't gain six kilograms. It's because as soon as that energy is generated, it's used. And so there's no Tesla battery that sits in our bodies that we can store that energy in and use it as a reserve. So mitochondria are on 24/7 your entire life. If they're off, this leads to dysfunction. The heart becomes a conduit for this, right? This is where you constantly have to beat in order to survive because you got to get blood to all your organs. And the heart beats all the time until you're literally from the day you're born till the day you die. That 30% volume translates into unique aspects. So if we zoom into a myocyte, which is the muscle part of the cell of the heart, it is this boxcar looking shaped cell. It has this amazing architecture. We do lots of isolated cell studies in the lab, and if you were to isolate this cell from an organism and put it under a microscope, you don't need to, you can actually see it without a microscope. They have these long boxcar-like shapes. You can see the striations in it, and it's just a beautifully looking cell. If you look at the mitochondrial density in that cell, there's three places where it exists in these unique platforms. There's something called the fibers inside the muscle, and so there's a lot of mitochondria across those fibers. This is the powerhouse. This is what's making all of the ATP that the heart needs to function and do the beat-to-beat kinds of things that it does. But then there's two other populations that don't make energy. They do other things. There's this set of mitochondria that sit right below the cell surface, so right under the plasma membrane. These are called the subsarcolemmal or the submembrane mitochondria. Their main role is to sense the environment, right? So they're sensing signals that come in and out of the cell, and they're allowing the cell to then respond to that signal in a positive or negative way. And so mitochondria outside of the powerhouse become this gatekeeper of cell death and survival dependent on the stimuli that they're sensing. And then there's this third population that I think people are starting to think about that sit around the nuclei or the nucleus in the cell. And these are called perinuclear mitochondria. And so the other big thing that mitochondria do is they dynamically shift in a cell. So they're constantly moving and they're constantly being degraded. And so mitophagy and autophagy are big things that are involved in resiliency, aging, longevity, and mitochondria have to die, right? You have to recycle these and reabsorb the aspect. And so not only do they create energy and create the signaling potential, but then they go through this death, dying, decay cycle, and this is a critical feature. And so I think they're really starting to emerge as more of global players in a cell that do a lot more than just make ATP.
Hmm. Yes, because I know we speak of mitochondria as athletes or exercising of exercise metabolism and not realize, oh, they're not just in our muscles to help create fat or carbohydrates for fuel source for my exercise. And there's a lot more of
Yeah,
them in our body, but we get so narrow-minded, I think, and don't realize, oh, the rest of the body
yeah,
is huge. Now, with the also the skin health too, I know I keep getting emails from the mitochondria in our skin help with aging. So you're just saying they're underneath the layer of our skin as well. So when we're trying to improve how we age, skin health, gut health, brain health, muscle health, it's all correlated to the role of the mitochondria in keeping them healthy.
yeah. I mean, it's energy, right? Everything in our life is tied to that energy balance. When you make enough of it, everything survives and does really well. When you don't make enough of it, things start to decline and you have to make choices biologically. And so if the heart can't make energy, it has to decide what to do. If the brain can't make energy, it has to shut certain systems and things down. And so that's sort of the big aspect of it. And then a key feature to this is oxygen, right? Oxygen, this is why they call it mitochondrial respiration, because your mitochondria are breathing that oxygen and they're using it to create that energy. In a perfectly tuned system, the oxygen basically turns into water and energy at the end of the day. In a suboptimally tuned system, that oxygen becomes a free radical and starts creating lots of injury in that system as well. And so it's this constant drive that you have over and over and over again. And the crazy thing is life doesn't stop, right? And if it does, you're dead. And your mitochondria are key to that. They keep moving you along in this continuum.
Mhm. Yes, I pulled out my book. I have had a while now, the mitochondria book by Dr. Lee. No, I think it is. And so you really go deep dive science and how complex the mitochondria are and the reactive oxygen species you mentioned and how we need that balance. It's same thing with insulin and cortisol. They're not bad things. We need them, but just the Goldilocks effect
Right.
seems to come into play for everything. So we
Yeah.
don't have excess oxidative stress as we age.
Yeah. And I mean, they do lots of other things in a cell, right? They buffer calcium, which is one of the only questions, one of the hardest questions I think in physiology they got wrong back in grad school was which ion is the most highly regulated in a cell in terms of what's outside versus inside. Turns out it's calcium. The cell uses calcium to do critical things, like your brain uses it to release neurotransmitters and other things. But you can't have a lot of this calcium sitting around in that cytoplasm because it's going to do negative things. And so the mitochondria become a huge sink for calcium. They store it and they buffer it in this unique kind of way and give the cell this reservoir of where to release and move things around. They're involved in thermogenesis. I mean, there are certain organisms that have a lot of brown fat. We try to get brown fat, but as we get older and older, we lose it. It's typically on our back. So one of the questions, or one of the things a lot of people do, will take cold showers, and they'll usually let it hit on the back of their sort of neck kind of thing, which is where that brown fat typically is. And its main role is to create heat, right? It just burns energy in the process of uncoupled state, and you just make a ton of heat in response to this. And it's a metabolic trigger. It allows us to burn a lot of fat very rapidly. Um, there's certain organisms that do this, and I think they have these adaptive advantages. Hibernating organisms manipulate their mitochondria in a unique kind of way as well. So they really do serve this global function of not just energy, but lots of other things in a cell that they regulate. It's a master regulator, essentially.
Mm. Yeah, it's a good word. Now, with the lactate research out, you spoke of something you didn't learn correctly, or myself too, in exercise physiology in college, but how things have changed is the role of lactate metabolism in the cell and how we can reuse that. Have you done any information on that correlating with the mitochondria
We
function?
have it. I mean, that's one of the things we're interested in looking at. So the closest test, or the most prevalent test that people have used outside of mescreen to measure mitochondrial function is this organic acid test, this oats test, right? And
Hmm.
one of the four things, I can't remember what the other three are. I know they look at lactate in your
Hmm.
urine as an
Hmm.
indicator of mitochondrial health, and I think there's like three or four other metabolites that they're looking at. So lactate does become an important feature. We haven't looked at this, but it'd be an interesting thing because as you exercise, you're creating a lot of lactate, and whether it gets recycled or not could be an indicator of how resilient you are ultimately, right?
Isn't that too? I heard Dr. Ben Bickman was doing a video on that and how your brain, if you have high levels of lactates, no more a sign of maybe insulin resistance or poor glucose use for your brain as well as your
Yeah.
heart.
Yeah, there's
Well,
a guy at Scripps, I can't remember who this was. Maybe it's the same guy, but this idea that Alzheimer's is actually type 3 diabetes, right? It's
mhm.
this inability of your brain to utilize glucose. And that's the other aspect of this. Each of our organs uniquely uses fuels in a different kind of way at different stages in our life. The brain is actually a good ketone user when you're really young and developing, and it switches to a glucose-based metabolism when you're older, which is why your brain is so sensitive to stroke and other things. If you deprive it of the blood and nutrients and sugar that gets there, it doesn't do very well. Whereas the optimized heart really moves from a neonatal stage, it's likely using glucose as the primary fuel state. As you become an older individual and the heart matures, it starts to use fat as a fuel state. But then it's interesting how you regress when you have disease. So in a heart failure model, the heart then switches away from the fatty acids to then starting to use glucose again because it needs to stabilize and maintain and adapt, right? And so fuel becomes a critical feature. And so, I mean, the simple thing to live well is to move, eat, and sleep. And it's to eat the right things, but it's so hard to do that, I think. And
Mhm.
so fuels become really interesting.
Mhm.
We've been looking at this with Sachin Panda and others for a long time now, is this idea of time restricted eating and could you vary diets in unique ways to create a stable fuel state in different systems, right? And I don't know. I think the
In
jury's
people or
still
fruit
out.
flies?
Everything. We're doing people. We're doing lots of mouse studies, lots of people studies, and all kinds of things around this space. So,
Hmm. Going back to cognitive health, I've been going down a lot of research after talking to Dr. William Davis and Martha Carlin. We've been going into the role of cellular health for Parkinson's, specifically with Martha Carlin, but also cognitive decline as you're speaking of strokes. I just met a friend last weekend who's late 50s and just had a stroke. And, looking at why, I always ask why. How does this happen? What's
yeah,
going, wrong with people as we age that these are happening,
yeah.
areas are happening so much earlier.
So we have sort of two ends in the lab. I'm a cardiovascular biologist, so we've always focused on the heart. And then one of our first postdocs, who's now a full professor and doing amazing stuff, we don't want to compete. And so I said, you look in the brain, right? And so he
Hmm.
developed this protein and all these things in the brain. And it's gone amazingly. We're actually going to be in a clinical trial, hopefully by the end of this year, for a target for ALS. And so the neurodegenerative aspect becomes really important. But we've had these, and we collaborate very closely. And we have two ideas around this, right? Is the heart more important or is the brain more important for an organism to live? And we've created this protein that we've been interested in, which ties back to the membrane called caveolin. If you make more of this protein only in the heart, and so you basically create a transgenic animal, which you can do genetically and modify it, this animal lives really well, right? It's resistant to an ischemic injury, it's resistant to heart failure, it's resistant to diabetic cardiomyopathy. If you age them to 2 years of age, which is comparable to an 80-year-old human, they function from a heart perspective like they're 3 months old. It's like they've never aged. Same thing, if you express this protein only in the brain, they cognitively live really well. And so at 2 years of age, they're performing mazes and other things like they're 3 months old. So they don't age. So the membrane becomes this huge conduit. One of the things we've been arguing over is, well, what's more important then? Is it the brain or the heart? And the pump becomes a really important thing. If you don't pump blood to the rest of the body, the brain doesn't matter. You're going to die anyways, right?
Mhm.
And so it's really this idea of resiliency in your cardiovascular system. There's more and more and more evidence emerging that these neurological diseases that people have, neurodegenerative diseases that individuals have, their primary cause is likely cardiovascular in nature, that you have this compromise of the blood-brain barrier, that you have vascular abnormalities that then ultimately lead to this dysfunction in neurons and other things long term, right? And so
Mhm.
I think there's this interesting interplay between the heart and the brain that happens in a unique way. A lot of us focus on sort of muscle health, but I think it's really heart and brain health that we need to focus on.
Side note, I've been diving down another rabbit hole, which I tend to do more and more lately. The role of the vagus nerve in that too,
Yeah.
functioning. Is there any correlation with poor vagal tone and heart health and how that could somehow impact the mitochondria? It seems like I always come down to how does the
Yeah,
mitochondria
I think,
play
I
a
mean,
role in vagal?
I think there is, right? The vagus innervates the heart and it innervates the gut. And so I think that
Hmm.
the microbiome speaks to everything else and it's the brain that's sort of masterminding this, but it's really the microbiome that's creating this conduit. So there are people on
Amazing.
campus that are creating these vagal nerve stimulators and there's a vagus nerve society and all these things that are out there
Yeah,
that
I
are
just joined
pushing
it.
this idea. Oh, you did. So is this with Mark Tagger and some others?
I just joined, but I've been working with Nuvana. They do a vagal nerve stimulator, and Amy, the
Okay.
co-founder, sent me a link to sign up because I'm so geeking out. It's
Okay.
all
I mean,
together.
I think there's a whole idea around this. One of the things
Mm,
that we're seeing on this mescreen test is the individual that's over hacking, right? That's overdoing the exercise, overdoing the supplements,
mm,
overdriving. They think they're going to live forever because they think they're doing all the healthy things, but they're actually exhausting their mitochondria.
yeah.
And I think the better thing to do is to figure out how to make your mitochondria rest when you're at rest and to
Mm,
do absolutely nothing, and
hmm.
then have them really become activated when you need them for a stress response or an exercise or something like that, and then really come back down to that zero baseline very quickly. And so most people are interested in the peak that they can drive. I think one of the things we're interested in physiologically is not the peak, but how quickly you come back down to resting,
Mm,
which I think is the better measure of how adapted you are. Because if you think about it, right, things that live really long are ones that don't, not that they're couch potatoes, but they're ones that don't really engage much with around them, right? The
yeah.
longest lived organism on planet Earth, any ideas?
Mm, no. I'm trying to think.
The giant
Uh,
clam, right? It's this
oh,
gigantic thing that has this hard shell, and so it can completely limit its outside from its inside.
yeah, like a
And
turtle, same thing, right?
yeah,
They have
I
a
mean,
shell.
500 years is how long this thing lives. And so this idea of resting and digesting, which is all the vagal nerve stuff,
Mm-hmm.
tied to this idea of a basal, we call it almost like this hibernative state, right? You go into the stasis, and then when you need it, you just come out of it like nothing happened. And there are these studies that people would do on hibernating bears and stuff, and it's sort of an interesting correlate, right? These bears and other organisms go into this hibernative or torpor stage where their system looks like it's dead, right? They're
Mm-hmm.
completely shut everything down. Their metabolism goes to nothing. And then just like that, they wake up like nothing's happened and they walk as if everything's great, right? That
Mm-hmm.
that ability to understand that hibernative process and how they reanimate, I think is going to be key to all of these things that are discovered about longevity. And not
Yes.
many people are thinking about it, right?
Yeah. Well, I always say more is not better, less is more, the Goldilocks effect, and acute stress versus chronic stress. And that's what I would say. You could be doing all these good hormetic stressors that
Yeah.
are biohacks, but as you mentioned, too much of everything all accumulates, I would say, in a beaker of stress. It just comes into one filter. So if you have a zillion different stressors, you're not giving your body that rest time. And so
Yeah.
it's an important part to look at that. So going into some definitions and then getting to your scan, mitochondria we went over, but just brief, what comes to mind when you say biogenesis?
Yeah, so mitochondria are destroyed, but they're also created, right? And so the biogenesis is that aspect of making new mitochondria. And there are proteins and master regulators that have been described. PGC1 alpha is one of the
Mhm.
things that most people think of when they think of mitobiogenesis. This is one way to track it. There's also ways to look at mitochondrial DNA content. So it turns out it is another organism living inside us, right? And it has its own DNA. It's this plasmid circular DNA. It has a specific subset of genes that it encodes for, and these are part of the electron transport chain that move these electrons in unique ways to make energy. And so a couple ways to track PGC1 alpha, mitochondrial DNA. The other hot area that a lot of people are looking at right now is that they think that mitochondrial DNA actually circulates in our bloodstream. And it may be
Yeah.
an indicator of stress responses, that
Mm.
when your cells are starting to die and decay, they release the mitochondria, and then this gets floating around and it creates pathology. And so there's lots of ideas around that as well. So,
Hmm. And then the opposite of creating new or getting rid of the dead. So
yeah,
autophagy, but mitoautophagy.
yeah.
And how would you do that?
There's a couple ways to do it, right? Mitophagy can be activated. A couple supplements that are out there on the market tend to do things like that, like urolithin and things along those ways. Intermittent fasting, which works in a subset of individuals, I think about 65% of individuals typically respond.
Hmm.
That is a way to activate autophagy, mitophagy processes. So as you do this cycle of starvation and feeding states, your mitochondria in that starved state start to decline and they'll actually self-ingest. I mean, autophagy is basically self-eating, right? And so you're eating yourself.
Pac-Man.
Yeah, so you're creating these raw resources back into the living system.
Yeah.
And this is a good way to turn things around. There are proteins that signal this, and this is what we think goes wrong with aging and disease is that these targeted pathways that
Mm.
regulate autophagy, mitophagy go awry, that you have, either mutations in these pathways or these pathways are no longer working the right way, and then you just get accumulated mitochondria and more damage. The story in the heart has been sort of biphasic and different. There's some people that suggest that it's tinier mitochondria in your heart that lead to this aging phenotype, and others suggest that it's giant mitochondria that can't undergo fusion fission reactions and continue to just get larger and larger that create this. No one really knows yet, right? And so these are sort of living dynamic processes that are hard to capture.
Mm. Yeah. And what about AMPK and AMPK versus mTOR? To AMPK, we talked about recently
Yeah,
on the
I mean the
mitochondria
the thing that a lot of these biohackers are on are metformin, which would be sort of that AMPK target.
or berberine.
Yeah, there's evidence for this. I think there's again, I think there's this notion of certain things work for certain people and other things don't, right? And so I think it's really, you have to know what your body physiology, body biochemistry is and how it works. Mescreen gives you a unique look into that biochemistry. One of the things that I think gives us an advantage, so like these mTOR pathways, the feasting, fasting things, will give you activation of mTOR in these things. The other thing that hackers are doing are rapamycin, which would target that mTOR pathway. Um, it doesn't work for everyone, right? And it may be toxic to a lot of people, but no one's really had a way to gauge this. One of the things we're positioning mescreen as is this is a new vital sign, and it really gives you this way of looking at all of these hacks and lifestyle kinds of things that you're doing. So the idea is to do a baseline to get a sense of where you sit and then start a journey, right? Whatever you're going to do, whether it's lifestyle modifications, diet, exercise, or go into supplements or pharmacological agents. You can then be on them for a certain number of months and then retest to see if they're actually doing what they say they're doing. One of the first things we did when we were sort of validating this test is I bought the most expensive mitochondrial supplement on the market. I can't even remember what it was. We had a couple people in the lab try it for a month, so we did a pre and a post. It did absolutely nothing,
I
right?
heard that. You
So
say
there's
that.
so many things out
Yeah.
there. If you go to these biohacker events and stuff, you can walk maybe 20 feet and spend $15,000, and most of those things will target and they will claim mitochondrial functionality. But there's no evidence to support any of that, right? And so our test gives the person the power to figure out if that 15 grand they just dropped on 20 feet of space is actually
Hmm.
doing what it claims it's going to do, right? So,
Yes. And that's why we say test and not guess as FDN practitioners, and why I'm all about nutrition, the exercise, the lifestyle habits, the stress and the sleep optimization is key. Then supplement where you need to, and then retest to see, are you doing everything to make a change? Are we seeing a
yeah,
shift positively, or do we need to course correct, stop, push, pause, and recalibrate, remeasure because you don't know what you don't know?
right. True.
And with
And
exercise,
everyone wants an easy answer, right? They just want to
yeah.
just give me a pill. I just want to take a pill that'll fix everything, and it's not that easy. And that's the
Mm-mm.
problem I think with us in life is there's very simple answers to things. Most of us don't want to do the simple because it takes a lot of effort.
Well, that's what I'm always laughing about. That I look at, how are we going to age? Is recreating our current self. So we're our best self in the future. As I watch my mom, who's 83, my dad passed at 80, and seeing what's happening to all of her friends and Parkinson's and muscle health and injuries and liver health. And
Yeah,
so that's why I kind of shifts my focus on the podcast to help. Let's do what we can after age, 40, but 45, you should really be focusing on being a health optimizer, taking ownership of your health today if you want to be thriving and being in your prime when you're 70, 80, 90
I agree.
years
I mean,
old.
I think there is a too late to start model in this, right? I think we've seen this in animal models and things. When you try to reverse decades, years of dysfunction, it just doesn't work, right? I think
Mm-hmm.
you have to create a modality that you start and then you maintain that throughout a lifetime.
Mm-hmm. Yeah, for sure. So I want to share the screen and go through the test. Now, as I talk often about metabolism and how to speed up metabolism and the role of mitochondria, if we do resting metabolic rate, for example, we can identify if you're a good fat burner, fat oxidation, and that's with the mitochondria and AMPK, versus if we do a test and your metabolism total amount of calories a day is slower than it should be if you're height, weight, age, gender. Do you need to speed up your metabolism? That would be mTOR, right? And doing more strength training and protein. So it's just, I would say it's not one or the other. We need to optimize both and keep
Yeah,
them in check. And then exercise intensity, how well do we use fat? Oxidizing fat happens in your mitochondria versus as we go to that metabolic crossover point, we start to see a shift in more carbohydrate dependency and then higher intensities, different fuel sources. But the role of fat oxidation is always so fascinating to people, peak fat burn. And I think a lot of people are inefficient and need to know, are my mitochondria healthy as the test you're doing? And then how can we see in movement or at rest?
yeah.
All right. So let me share your website so people can learn. I was just doing this in the beginning. So walk us through the test if we see
Yeah, so
here.
basically you get a kit to your home, or we're selling this to a lot of functional medicine providers and functional medicine clinics as well. So if you go to that
Yep.
clinic, they'll have a test for you. You'll register your kit, and then you basically go and do a questionnaire. So in the questionnaire, we're trying to capture demographic data as well as Promise 29 survey kinds of things. So we want to see a community do this. So as more and more people take this test and feed us this data, we'll be able to give you some metrics of where you sit relative to someone in terms of their age, their sex, disease, normal, completely endurance athlete, and all these kinds of things. And it's a way to gamify this ultimately, right? To give people more feedback into where they're sitting and how to really optimize their mitochondrial health. So inside the kit, you have all the things you need to collect,
Mm-hmm.
all the things you need. So it's a two-window blood card. And so one of the things that we struggle with in this is most people are used to dry blood cards, right? So they'll basically put a spot on there and they'll be done. This is a little more advanced than that. It's a bilayer system. So you actually, what you do is you poke your finger and you saturate that small square at the top. As that saturates, the blood starts to move across those three lines in the next window. And
Uh-huh.
as it's moving, it's separating the serum from the red component. And we're interested in the serum, is where all
Uh,
these factors are that we're using in our assay. And so you basically, we only need one window, but it's great to have two just as a backup or other things come up. And so you fill the first window, fill the second window. You let this sit for a little bit of time to dry out. Once it's dry, you put it into a desiccated pouch, put it in the mail, send it to us. When it arrives at our facility, we then extract that serum and then it goes into our assay platform. So then
oh,
usually,
but I just wanted to throw in I
yeah.
listened to you on different interviews on podcasts, and isn't it some special room that you do the data collection? It's some sealed off room to when you're looking at the
Yeah, so we have
results.
the facility is moved off site, and it's a first, I guess, in the world mitochondrial lab testing facility, and so it's a special bio room that you have to be gowned up and do all this stuff in. The reason why this test is expensive and is very, I think, just not the right word, very sort of expensive, but then gives you some deep insights into your mitochondria, is we're using a living system to test this, right?
Mm.
And so we have to culture these cells all the time, and it has to be a completely clean facility. We have lots of these cell culture facilities on campus that we work in. My main one is a couple doors down from my office. And we had a fungal contamination that someone brought into the facility last year. It took us 3 months and 10 grand to clean up the fungal reaction. And so we're hypersensitive to who walks into what space, what
Mm.
shoes you're wearing, what clothes you're wearing. And so you literally have to take your shoes off, you put Crocs on, you gown up so that nothing is exposed to that space. You go, so it's a very clean facility that you're walking into. Um, so what we do then is it's this actual living cell that's sitting on our system that's measuring oxygen and extracellular acidification to get a sense of mitochondria and glycolysis. So we then take the biochemistry that you're creating in your body that we capture on this card, we move it onto this living cell completely, and then we start to see how that cell responds to your biochemistry. And
Mm.
so the first thing we do is what you essentially talked about is what's the basal change in that cell's metabolism. And so if that mitochondrial respiration goes up or down, it tells us that there either is something in that biochemistry that's hyperactivating or hypersuppressing that mitochondrial functionality. And then we essentially mess with the mitochondria in that dish through different chemicals. And so we can test the efficiency of your mitochondria. We can test the exercise potential of your mitochondria and how much reserve they can generate. The energy balance portion of the test looks at the same sort of setting and same setup, but looks to see under basal conditions, are you making energy with mitochondrial glycolysis? And is there a balance shift in that? We see unique patterns in certain individuals. People who are energy compromised tend to be more glycolytic than their mitochondrial. People who are optimized for sort of exercise and endurance, they tend to be more mitochondria. And we're seeing a new sort of profile emerge. We see individuals that I call hibernating, like amazing people that live well. They actually shut all of their metabolism down basally. So they have a really low mitochondrial and a glycolytic score, but they have this really high potential. And so what we were talking about earlier, these individuals that at rest and digest are almost like they're dead on the system. But then when you challenge them, they're able to create a huge energy reserve. And so the test will give you elements of that. And then the two final things we look at are reactive oxygen species generation. So this is a different type of assay where we look under basal and stress conditions, how much reactive species you're generating. And then the final component is your mitochondrial network, this idea of the dynamics of mitochondria. We've actually made this special cell that expresses a green fluorescent tag only in the mitochondria. And so under our live cell microscope, the entire network becomes live. And then when you add your biochemistry, you can see how that network shapes and shifts and changes. And so we've created an AI algorithm that then looks at your network stability kinds of things. And so by looking then at your sort of stress response, your energy balance, your ROS, and your network, you really get this full picture of your functional and dynamic structural sense of what your mitochondria are likely doing in your body. And so it gives you a huge way to then think about your mitochondrial health status and how to possibly change it through lifestyle interventions and other things. So
So, couple of things before we move on to the step
yeah,
5. Definition for people if they don't understand mitochondria efficiency and the mitochondria fueling. I think you're talking about oxidative phosphorylation or fat metabolism versus glycolysis.
yeah. So the efficiency really is this leak state that we're measuring. So how efficient is that oxygen moving through that system? When we stop that oxygen, is there side places where it's shooting off and those kinds of things? So we can determine that. The energy efficiency or the energy balance really is looking at if you're using that energy with mitochondria or with glycolysis, right? We can sort of toggle the two things by looking at the oxygen versus the extracellular acidification, which is an indicator of glycolysis.
So when people are hearing mitochondria, you're only using fat for fuel when you're oxidative phosphorylation using fat, or some glucose in there too when you're looking at
Glucose
the
in there as well.
energy.
So the cell is optimized as a glucose loaded system. And so we're using it, but we can tell if that's being used in the mitochondria or in the cytoplasm and glycolysis.
Wow, really? That's fascinating. Okay. And then ROS generation, oxidative stress. So you can measure that. And
Yep.
that's where we want to make sure is not excessive as the Goldilocks effect again. So we don't want too little or too
Right.
much.
Yeah, and our reactive oxygen species are good, right? I mean, there's back in the day when I was getting my PhD, this is when nitric oxide and the biology around nitric oxide was discovered. And it was this notion that there's this free radical that actually creates a huge impact on vascular biology. And nitric oxide has turned out to be an amazing molecule for
Mhm.
both signaling as well as death and decay as well. If you have too much of it, you create injury, but if you have just the right amount, you create a lot of signaling potential. And this is what Viagra is based on, right?
Mhm.
And so, same thing with mitochondrial derived reactive oxygen species. A little bit is very good for signaling, too much then gets into the damage potential.
Hmm. Okay. I'm gonna remember the other thing that just popped in my head too, because your focus is cardiology, of how this relates with heart health and the whole study that came out last couple days ago on APOB markers and LDL and how cardiovascular health, this could play a role in cardiovascular health, looking at your mitochondria function in your heart
Yeah.
specifically. Can you tell that?
Yeah, so we have optimized to muscle right now for the mescreen launch. We think the skeletal muscle is a global capture of someone's metabolic state.
Mm-hmm.
But with the NASA design and things we've sort of articulated in the discovery phase, we can eventually launch a mescreen cardiac, a mescreen neuro, a mescreen liver, a mescreen kidney. We do a lot of disease modeling in the lab as well, so we could easily do a mescreen Alzheimer's, a mescreen ALS, a mescreen Parkinson's. And so there will likely be many offerings as we advance the technology.
That's exciting. Okay, so we sent our test results in 3 to 4 weeks. You get to the lab, you do your amazing study looking at all this data, and then the results come into a portal.
Yep. And then
And
you can
what do we
either
do?
access that portal if you're a consumer or if you're working with a practitioner, the practitioner
Mhm.
then gets access to that and they'll talk to you, right? They'll go over your
Mhm.
results. All of our practitioners are trained to think about mitochondria, and a lot of them have their own protocols they want to try on you. And so now you look at your mitochondrial results on the Me screen, you decide what the next engagement is with your provider, and then you do that thing for 2 or 3 months. You retest and you see if things work. If they don't, it gives you a next target of what to start switching and doing.
What do you find if, from experience, getting the results to improve them, focusing on non-supplements first, what would you say is helping?
I mean, the key feature is always diet and exercise, right? But to
Mm.
get someone to do that for long term is really, really hard.
Mm.
But intermittent fasting, we've seen really good results with. I think there's some evidence in the literature around heat and cold stress. Heat shock proteins are key features of that, so I think they do amazing things. Um, Outside of that, I don't know. I mean, there's supplements and things that are out there, mitophagy, autophagy activators that I think have potential. It's a
Exercise?
it depends on how you do it though, right? I was just talking to one of our consultants this morning around this, and she had recently seen a couple individuals that are hyper exercising individuals and their tests look horrible. And I'm like,
Mm.
maybe they're over exercising, right? If you're
Yeah, you
over
think?
stressing your system. And so, I mean, if you think about like the blue zones, which there's lots of problems with the blue
Yeah,
zones,
for
the
sure.
biology, but one of the things that people have noticed is the social interactions that people have. But then when you look at the exercise that they do, they're not in the gym like killing themselves to get their heart rate up and stuff.
Yeah.
They're in their garden gardening, or they're walking uphill both ways to wherever they're
Yeah,
going, kind
all
of
the
thing, right?
time. Stairs,
So
hills.
they're doing normal daily activities that keep them moving and active, and not
In nature,
necessarily
fresh air too.
right. Yeah. So I don't know if you have to get your heart rate up above a certain thing all the time.
Mm.
I think it's important to do these high intensity, short bout kinds of things, and then normal daily, just move a lot,
Yes.
right?
Yeah.
And do a lot of that. I think there's a feature to that where you're fat burning and you constantly keep that system churning and moving. As a cardiac person, what we see is when you do stress your heart a lot, you can get toxicity from that, right? There's this element of you're releasing catecholamines, beta receptor agonists, and other things in your body that then re-impinge on your heart, and they
Hmm.
actually cause death, right? So
Hmm.
there's ways to kill the myocyte, and overactivating is one way to do it. So
Mm.
it's a balance. And I
Yeah.
think you got to find what works for you. And for a lot of people, I think they tend to overdo it because this is this thought. If a little bit works for me, a lot is going to be even better. And it's
Yeah.
never that way, right?
No.
Everything, the Buddhist principle, everything in moderation.
Yeah. Well, that's so interesting because I see people at the gym, even this morning, that I thought this guy was doing sprint intervals in the bike. I'm like, great, he's doing 20-second sprint intervals, but he didn't stop. And I was doing stretching mobility on the floor nearby, 10 minutes, 15 minutes, and he's
Yeah.
still going.
Wow.
And I want to say, I don't know. Yeah, is that feeling good? It just didn't seem intuitively that that was healthy, but that's people's mindset to work hard. I need to do it this way and go 10, 20 minutes all out, which isn't
Yeah.
all out. And it's interesting when people hear, I keep trying to do videos on what is really HIIT training and what is sprint interval training that improves mitochondrial biogenesis is
Yeah.
not what people are doing.
Right. No, and I think one of the things that a lot of older individuals, once you're 40, you struggle with is muscle mass, right? And it's this resistance training to really keep up that muscle mass that's going to be key. Lean mass is what burns a lot of the glucose in our bodies. And the more of it that you have, the better off you're going to be.
Yeah. And do you see that with the mitochondrial health? I read sometimes that the strength training does help mitochondrial health.
It does. I mean, one of the problems I face is omental fat. I have a lot of it on my midsection. This is one of the problems I think with this. The more muscle you have, I think the better off you're going to be ultimately.
Mhm.
And it's this
Yeah.
constant battle. As you get older, it's hard to keep that lean mass on.
Well, I keep podcasting about that too. It's like, what do we need to do differently when we're getting middle aged that what we used to do doesn't work the same? And so, even I did a recording last night that protein to stimulate muscle protein synthesis, you have to eat double. And to
Yeah,
hit that leucine threshold, 2.5 to 3 grams now needs to be 6 grams for us females with low estrogen. And then to get 50 grams in a meal to make that work, which is so hard to eat that much for a lot of
it
us.
is. I think a good fat diet is probably better off than anything protein or like a mixed thing, right?
Yeah. I know. So I want to go over that on the website. One other thing you just said, the fat is talking about the cell membrane, and just I know we're going to run out of time, but the I was listening to this the cell membrane, the C15 and the fatty
Yeah.
15 that's out. How does that correlate with the mitochondrial health in your research and
Yeah,
testing?
so we're a membrane laboratory, and so if you look on all these hallmarks of longevity, they'll talk about 9, 11 factors that are key to longevity. In all of these, they're missing the most important factor. They never list it. It's the membrane. We were talking about the giant clam. The reason this thing lives to 500 is it has an amazing shell, right? Which is its membrane. It's able to limit the outside from the inside. And so we really think adaptive systems, if they can limit what's the outside stressor from the inside soft environment, you get longevity. That's how the system is built to survive. It's if you look at all chronic diseases, I think they're primarily a membrane disease. It's when the outside starts to impinge on the inside that you have cardiovascular dysfunction, that you have Alzheimer's, you have Parkinson's. All of these things, I think, at the core start off as a membrane dysfunction, which then leads to energy compromise and other things. And so at the heart of all of this, literally, is I think lipid supplementation. I think getting good lipids into your body that aren't floating around in your blood, but actually incorporate into your membranes is a good way to create resiliency long term. And C15 we haven't done any tests on, but there may be some things around this. But there are cheap supplements you can buy, phosphatidylcholine, serine, that would be a great way to build that base membrane.
Mhm. I'm laughing because I think they're by you. It was a podcast with Dr. Mark Harmon on his podcast show about the dolphins and C-15 in San Diego. They did a whole study. The Navy has
Yep.
dolphins, and
Yeah.
they did that study on it. So it's like dolphins, and that's how they started to learn about the C-15.
Yeah, it's a
So
company I think based in San Diego that's doing it.
yeah. So if you need to collaborate doing
Yeah,
me
yeah. I
scans
know we
on
have
the
some
dolphins.
connections to them, and so we're thinking about it. So we'll see where that ends up.
Well, I know you have a busy day, so where can we send people to the Me Scan website to order is the best, probably, I
Yep. So
assume.
yeah, the mescreen website. It's mescreen.com that you can sign on to, and then we can come up with a code that gives you a discount as well if people do it through your podcast.
Yeah, I usually say Coach Debbie or Coach Debbie Potts. We'll look that up. Yeah, I love this stuff. Love what you're doing. And I know, you talked to the same week I was talking to you, the guys in Encinitas here at Luminary or Illuminati.
Yeah.
They're
Yep.
doing all they're doing and see how we can test and put all this together. It'd be really fun.
Yeah. That'd be great.
See how we can improve how we age is what I think we're all focused on, health optimization.
Yep.
So thanks for your time today. Love what you're doing and keep in touch with any new updates.
Yeah, it's great.